Okay, so today's unit is biochemistry. So biochemistry is a large unit that can be broken down into the following subtopics. We've got molecular biology, carbohydrates and lipids, proteins, and water. But since it's such a long topic, today we're just going to be talking about the first two subtopics, molecular biology and carbohydrates. So, molecular biology explains living processes in terms of the chemical substances that are involved. All living things are carbon-based life forms, and this is due to carbon's unique ability to form four covalent bonds that allow a wide variety of stable compounds to exist. This is also because of where it's found in the periodic table, group 14, meaning that it's got 4 valence electrons, so it's neither more inclined to attract electrons or to get rid of electrons. And these some of the very important stable compounds that are formed from carbon are carbohydrates, lipids, and proteins, which we need to survive. Biological activity is regulated by enzymes, which are expressed by gene activation, and metabolism is the sum of all enzyme-catalyzed reactions in the body, both the anabolic and catabolic. Anabolic reactions have the purpose of synthesizing complex molecules from simpler ones, using energy to construct the new bonds. The nature of these reactions are condensation reactions, as seen by the reaction of forming a glycosidic link between two alpha-glucose molecules, which is seen here, which produces water when formed. As you can see, when there are two alpha-glucose molecules, to form a bond, the hydroxide on one molecule and just the hydrogen from the hydroxide on the second molecule will bond together to form H2O, water, and then the remaining oxygen will be used to form a bond in between the two carbons. This makes them reduction reactions. Conversely, catabolism breaks down complex molecules into simpler ones, which releases energy when the bonds are broken. These are usually oxidation reactions and they involve hydrolysis. Bringing back the example from earlier, to break back down the bond between the two monomers of the disaccharide, water is required to replenish the hydrogen and oxygen released when the monomers originally bonded. So by replenishing the hydrogen and oxygen, we can bring this disaccharide back into the monomer state. Another important part of this unit is the theory of vitalism. So vitalism is the theory that all living things have a special life force that created the organic compounds, and thus they have a special non-physical element. This is a false theory, and it was proven false by the synthesis of urea, which is an organic molecule, from inorganic molecules in 1828 by Frederick Wöhler. He achieved this by heating an inorganic salt, which is here, ammonium cyanate, and that formed urea, which is a human waste product of nitrogen metabolism. And this showed that organic molecules are not fundamentally different to inorganic molecules. Now, there are 3 main classes of carbon compounds: carbohydrates, lipids, and proteins. And they are organic compounds that contribute very much to the function and structure of a cell. So we're going to be discussing carbohydrates in this video. So carbohydrates, they are the most abundant organic compound in nature, made up of C, H, and O atoms in this pattern for alpha glucose, which is there is an oxygen here and 6 carbons. And on each of the carbons that are in this ring structure, there is a hydrogen atom and a hydroxide molecule. And it's a pretty regular structure, except for the third carbon, which has a slightly different structure where the hydroxide and the hydrogen are flipped. So the hydroxide is on the top and the hydrogen is on the bottom. And this final sixth carbon is attached to the fifth carbon outside of the ring structure. In this way. And not to forget this final right here. And beta glucose has a very similar structure to alpha glucose, but it's just slightly different in terms of the orientations of the hydrogen and the hydroxide molecules. So here, actually, the first carbon also has a flipped hydrogen and hydroxide. So the hydroxide here is on the top and the hydrogen's on the bottom. So as you can see, they each carry 6 carbon atoms, 6 oxygen atoms, and 12 hydrogen atoms per glucose molecule, regardless of whether it's alpha glucose or beta glucose. And the common ratio, or empirical formula, as it's called in chemistry, is CH2O. And the N refers to just however many times you multiply it by. It will always remain in this ratio of 1 to 2 to 1. And one glucose molecule is C6H12O6. So the functions of glucose is they function as a source of energy, and they are mainly for short-term storage, meaning that they are the first to be used up when energy is required. This is due to their solubility in water, as we are very water-based life forms, which makes them very, very easy to transport in the bloodstream, as they don't need additional support to be moved around. And it also makes them very easy to be digested and utilized. They also function as structural components as part of RNA and DNA, but that will be discussed in another video. Single units of carbohydrates are called monosaccharides, mono meaning one and saccharide being the general term for carbohydrates. This includes alpha and beta glucose and ribose, which is another type of monosaccharide, has a slightly different structure. Here it's actually a pentagonal structure as opposed to glucose's hexagonal structure. And as you can see here, it's also got different orientations to both the alpha and beta glucoses. Most monosaccharides form ring structures, and they can exist in different 3D configurations called stereoisomers. When two monosaccharides bond together, they form a disaccharide through a glycosidic link, as we discussed earlier. And there are 3 main monosaccharides. We've got glucose, we've got fructose, and we've got galactose, which are the 3 main ones that you have to know for the IB. And depending on how they are bonded together, they can form different disaccharides. So 2 glucose molecules actually form a disaccharide called maltose, while 1 glucose plus 1 fructose molecule forms a disaccharide called sucrose. And one glucose and one galactose molecule forms a disaccharide called lactose, which is the sugar formed in milk or other dairy products. When adding 3 or more monosaccharides together, that is how a polysaccharide is formed. And there are 3 common polysaccharides which you need to know for the IB, and that is starch, which you may remember from GCSE, cellulose, and glycogen. Starch is found in plants, and that is how plants store their sugars for long term. Cellulose is found in the walls of plant cells, and glycogen is found in people. So there are some differences between monosaccharides and polysaccharides, which you can see here. So monosaccharides are actually crystalline in nature. When you look at a monosaccharide, it looks crystal-like when you look at a single granule of sugar, for example. Whereas polysaccharides are non-crystalline. For example, if you look at cornstarch, it's white and powdery. And as well as that, monosaccharides have a sweet taste if you eat them, like sugar, and polysaccharides are not sweet. Monosaccharides are also not very storable. They are very easy to dissolve and to digest, so they tend to be instantly used up as energy. Whereas polysaccharides are better for long-term storage because it takes time and energy to break them down into usable pieces. The type of polysaccharide can depend on the subunits involved and the bonding arrangements between them. So as we stated earlier, the main polymers that you need to know are cellulose, starch, and glycogen. So as I said, starch is for storage and it's found in plants and it's made of alpha glucose monomers bonded in a 1,4 arrangement mainly. However, starch is actually made up of 2 different types of polymers. You have amylose and amylopectin. Amylose is made up solely of these 1,4 alpha glucose bonds, and amylopectin is made up of these 1,4 bonds and some 1,6 bonds, roughly 1 every 20 subunits. Now, what these numbers, 1,4 and 1,6, actually refer to is that when we draw a glucose molecule, you can actually number off each individual carbon. So starting from the oxygen in the top right, if I can just draw a quick rough molecule here. Starting from here, which is the oxygen, the carbon that would be here is 1, and then going around 2, 3, 4, 5, and then the one branch to it is number 6. So a 1,4 bond means that this carbon right here would be bonding with this carbon here in another glucose molecule. That is a 1,4 bond. And a 1,6 bond means that the carbon at the very top of the ring structure, the one that is attached to the 5th carbon, would be bonding with the 1st carbon of the structure. So because of this 1,4 bonding arrangement, actually, it causes the structure to curve because if you remember from our diagram earlier, when these 2 carbons actually bond together, when these 2 carbons actually bond together, there are 2 hydrogens left attached to each carbon. And these 2 hydrogens are actually on the same side of the carbon. They both stay on top of the carbon. Now, hydrogens actually have a tendency to repel each other, and that has to do with a topic you will learn about more in depth later when we are discussing water, which has to do with polarity and slight charges. But in essence, both hydrogens have the same slight charge, and they're both positive. So they actually repel each other because like charges repel. So the hydrogens will try to arrange themselves in a position where they are the furthest possible away from each other. Now this causes the two carbon atoms, rather than staying like this linearly, they will actually bend away from each other slightly. So rather than being in a straight line, it'll look more like this, where it is just slightly bent. So the hydrogens can be away from each other. And so eventually, all of this comes together to form this helix-like spiral structure. And as for amylopectin, when the 1,6 bonds are formed, it actually creates lots of little branches branching off. So it doesn't actually get as kinked as the ambulose, and instead it forms a tree-like branchy structure. Now, the next one that you have to be aware of is cellulose. So cellulose is a little bit different than the other polysaccharides because it is made up of exclusively beta glucose. Now, that has some implications because beta glucose for the fourth carbon actually has a different order of the hydroxide and the hydrogen molecules. If you remember from earlier, beta glucose on this fourth carbon here has the hydroxide on top and the hydrogen on the bottom. But in this structure, the two hydroxides would be on opposite sides of each other. You see, like that. They're not next to each other. So as you can see, the beta glucose has the hydroxides on opposite sides and in opposite directions. And that's actually perfect because what happens is that it allows for this bond to form here, like this, but the hydrogens stay on opposite sides of each other. So actually they don't repel each other nearly as much as they do in the alpha glucose, and that allows for the structure to be very straight and rigid, as you can see. And thus cellulose is able to be supporting the plant cells because it's got a very straight and rigid structure as opposed to the curled and branched structures of starch. And another thing, it's made up of beta glucose. And actually, as human beings, we lack the enzymes that are needed to digest this. So when you ingest cellulose, it enters our body and it exits completely whole. It's egested completely whole as our body doesn't have the enzymes to break this down. And we actually commonly refer to this as fiber. Finally, there is glycogen. And glycogen is the only one of these three polysaccharides that is actually found in human beings. It's found in our livers, and it's our way of storing glucose for long-term storage. It's made up of alpha glucose, and it's comprised of 1,4 and 1,6 bonds. It's very, very branched, and although it may look somewhat similar to amylopectin, it can be differentiated because while amylopectin has about 1/1,6 bond out of every 20 subunits, glycogen actually has 1/1,6 bond every 10 subunits, which is twice the frequency. So it is a lot more branched looking than amylopectin, which allows us to differentiate them. And next, we will be moving on to lipids. I haven't done the lipids portion yet. Yes.